Development of the extraembryonic membranes represents an explosion of embryonic tissue growth prior to attachment. Mammalian embryos can be subdivided into two primary groups. In the first group, that includes most domestic animals, the pre-attachment period within the uterus is long, or in several weeks. During this time, extensive extraembryonic membranes form by a folding process that generates the amnion, chorion, and allantochorion. In the second group, or the primates, the blastocyst implants very soon after it enters the uterus. The extraembryonic membranes form after implantation or attachment. Extraembryonic membrane is a set of four anatomically distinct membrane originating from trophoblast, endoderm, mesoderm, and embryo. This membrane causes a dramatic growth of the conceptus due to its development. Its formation is an obligatory step in the acquisition of the embryo's ability to attach to the uterus of the dam. Furthermore, the extraembryonic membranes of the pre-attachment embryo consists of yolk sac, chorion, amnion, and allantois. The figure shown is the schematic diagram illustrating the typical development of extraembryonic membranes in mammals. This development sequence must occur before attachment to the endometrium can take place. The hatched blastocyst consists of the inner cell mass or the ICM, the trophoblast, and the blastocele. Very early in embryonic development, the primitive endoderm or the blue layer begins to form beneath the inner cell mass and grows downward, forming a lining on the inner surface of the trophoblast. At the same time, the mesoderm or the red layer begins to develop between the primitive endoderm and the embryo. When the primitive endoderm completes its growth, it forms a cavity called a yolk sac. This cavity does not contain yolk, but is so named because it is analogous to the yolk sac in avian embryos. The mesoderm continues to grow, forming a sac that surrounds the yolk sac and pushes against the trophectoderm, which is previously the trophoblastic cells. The newly formed mesodermal sac pushes against the trophectoderm and begins to fold upward, forming a wing-like structures called the amniotic folds. The mesoderm now completely surrounds the yolk sac and the developing allantois. The allantois is a diverticulum from the primitive gut that collects embryonic wastes. The mesoderm continues to fuse with the cells of the trophectoderm to form the chorion. The amniotic folds continue to grow upward around the embryo. The yolk sac begins to regress, but the allantois continues to grow and expand. The amniotic folds almost completely surround the embryo. The leading edges of the amniotic folds will eventually fuse. The amniotic folds have completely fused, resulting in the formation of a double sac around the embryo. The inner sac consists of trophectoderm and mesoderm and is called the amnion, which creates the amniotic cavity. The chorion completely surrounds the entire conceptus. The allantois continues to expand and begins to fill in the spaces of the cavity. Eventually, the allantois and the chorion will fuse, forming the allantochorion. The yolk sac continues to regress. The figure shown is the schematic diagram illustrating the typical development of extraembryonic membranes in mammals. This development sequence must occur before attachment to the endometrium can take place. The figure shows the interferon tau from the conceptus prevents luteolysis in the cow and ewe. So the interferon tau is produced by the trophoblastic cells of the blastocysts of cows and ewes. Interferon tau acts on the endometrial cells of the uterus to inhibit the production of oxytocin receptors so that oxytocin cannot stimulate PGF2 alpha synthesis. In addition, interferon tau causes production of proteins from the uterine glands. The arrows from the uterine glands indicate the movement of products that are secreted into the uterine lumen to nourish the conceptus. This figure shown is the estradiol reroutes PGF2 alpha to prevent luteolysis in the sow. So in the sow, the two major differences exist in maternal recognition of pregnancy compared to the ewe and cow. First, the conceptus of the pig produces estradiol that serves as the signal for maternal recognition of pregnancy. Second, PGF2 alpha is produced in significant quantities but is rerouted into the uterine lumen. The conceptus begins to produce estradiol between days 11 and 12 after ovulation. The production of estrogen does not inhibit the production of PGF2 alpha, but because the PGF2 alpha to be secreted in a different direction than in the cycling sow. The direction of secretion is away from the submucosal capillaries and toward the uterine lumen. The luminal PGF2 alpha has little access to the circulation, thus cannot cause luteolysis. The precise mechanism whereby the rerouting of PGF2 alpha occurs is not completely understood. However, it is believed that the estrogen causes increased receptor production for prolactin in the endometrium. Prolactin changes the ionic flux for calcium. This is thought to promote the exocrine secretion of PGF2 alpha into the uterine lumen rather than an endocrine secretion into the uterine vasculature. Porcine conceptuses produce interferons, but these materials do not affect corporal lutea longevity or function. Production of E2 or estradiol by the porcine conceptus not only serves as the maternal signal to prevent luteolysis, but also probably serves to stimulate contractions of the myometrium to distribute conceptuses with the proper spacing along the uterine horn. The figure shown is the transuterine migration of the equine conceptus. In the mare, the presence of the conceptus prevents luteolysis. Also, in the presence of the conceptus, endometrial production of PGF2 alpha is significantly reduced. A unique feature of maternal recognition of pregnancy in the mare is that the conceptus must migrate within the uterus from one uterine horn to the other. This migration must occur between 12 and 14 times per day during days 12, 13, and 14 of pregnancy in order to inhibit PGF2 alpha. This intrauterine migration of the equine conceptus appears necessary because the conceptus does not elongate in other species. Therefore, there is a less contact between the conceptus and the endometrial surface. In other words, the migration of the conceptus is probably necessary to distribute pregnancy recognition factors to the endometrial cells. At the left side, or the left picture, each black sphere represents a stopping spot in which the conceptus will spend between 5 and 20 minutes. The migration of the conceptus probably distributes pregnancy factors, which is represented by white lines over a white surface of the endometrium. At the right side, or the right picture, this uterus is from a mare at the day 14 of pregnancy. The uterus has been incised on the dorsal surface to expose the spherical conceptus. This specimen shows the conceptus and uterus on the last day or on the 14th day of the uterine migration phenomenon.